Okay. Which of the following bacteria, so bacteriology MCQs, which of the following bacteria acquires its toxin gene through specialized transduction by a lysogenic phage? Okay. So this is very high yield. You should be knowing. You remember we did bacterial genetics, talked about transduction. And under transduction, there was generalized transduction and specialized transduction. Okay. So here the answer is Corynebacteria, diphtheria. Okay. So there are certain types of bacteria that undergo toxic gene, that acquire the toxic gene through specialized transduction. Who knows the others? So the mnemonic is ABCDs. So I want you guys to write it down. So A is strep A erythrogenic toxin. Okay. Strep A erythrogenic toxin. So the mnemonic is ABCD, then ABCDs. So strep A erythrogenic toxin. Then the B is botulinum toxin. The C is cholera toxin. Then the D is diphtheria toxin. Then the S, which is ABCDs, the S is for Shiga toxin. I've also written it on the chart. Okay, so you have to remember that. Okay. So in fact, here the answer can be Corynebacteria and Clostridium botulinum, because we also said Clostridium botulinum toxin. Okay. Also Vibrio cholerae is there, so cholera toxin is also there. There are multiple answers. Didn't even see that. Okay. Then we go to number 2. The following statement is true regarding Staphylococcus aureus, except. Now, let's go through the choices. Choice A. Colonization rate rises in long-stay hospital patients. So this is true. In fact, we usually say when you stay in the hospital for a long time, you are at risk of getting resistant bacteria. So what that means is that these bacteria have mutated and they are resistant to the common antibiotics. So in this case, we have methicillin-resistant Staphylococcus aureus. Okay. Then choice B, all strains are sensitive to methicillin, so this is wrong. We have methicillin-resistant Staphylococcus aureus. We have vancomycin-resistant Staphylococcus aureus. Okay, so for MRSA, we give vancomycin. In case it is vancomycin-resistant Staphylococcus aureus, we give linezolid. The main mode of identification is by coagulase production. So if you remember, Staph aureus is coagulase and catalase positive. So for coagulase, how to differentiate coagulase? It is used to differentiate Staphylococcus, Epidermidis, and what is the other one? Yes, you should be knowing these.
Streptophyticus.
Staphylococcus saprophyticus, yes. So saprophyticus is resistance. Okay. And then responsible for community-acquired infection. That is true. Preformed toxins in food are responsible for food poisoning. And you have to note the word preformed toxins. Okay, so that's why when you ingest food that has Staphylococcus aureus, you will start diarrheaing and vomiting almost 1 hour or 2 hours after eating, because due to the preformed toxins. Okay, so I hope that is understood. And these are the enterotoxins. Okay, number 3, false about Corynum bacteria diphtheria. So they're asking which is the false one. Let's start. Non-capsulated, that's true. It has no capsule. Gram-negative pleomorphic bacteria. Is Corynum gram-positive or gram-negative?
I think it's gram-positive.
Yeah, remember we did them when we were doing the gram-positive. So I want you guys to know this. You should have that chart in your head where you know the gram-positive bacteria. Remember we classified in terms of coccus and rods, then the gram-negative bacteria. Okay, so I want you to go through that. Okay, so you have to remember all the time. So Corynebacteria is gram-positive, so it is not gram-negative. Does not produce spores, that is true. Spore-forming bacteria, we know it. Bacillus and Clostridium. And then we go to have metachromatic granules in their cytoplasm. That is true. Remember, we check that using the Albert stain. Easily discolorized during gram staining. That is true. Okay. Which species is frequently associated with nosocomial infections transmitted via medical devices inserted into the body? So this is a very direct question. What is the answer?
Is it D?
Staphylococcus epidermidis, yes. So I remember when I was teaching, I said Staphylococcus epidermidis is responsible for biofilm formation. It forms biofilms, especially on medical devices. So things like catheters, things like cannulas, okay? Because it is a normal flora of the skin. So once that happens, it forms biofilms and it can cause bacteremia once it enters the blood. And remember, you remember that? Okay, things like prosthetic valves and all those. The rest, no. Propionibacterium acnes, this is responsible for acne. Okay, then the rest, definitely not the answers. Then we go to the next question. In urinary tract infections, select one. Let's eliminate. Infection by E. coli is predominantly by hematogenous spread. So first of all, we have to know, how does a UTI happen? You get an infection via the urinary system. If you were to picture the urinary system from upwards downwards, it starts from the kidney, goes to the ureter, the bladder, then the urethra. Okay, so a UTI starts from the urethra as it ascends. Okay, so UTIs are spread via the ascending route. So it will ascend from the urethra as it goes to the bladder, then it can go to the ureter, then to the kidney, leading to things that we call them pyelonephritis. Okay, so it is not by hematogenous spread. Choice B, Staphylococcus saprophyticus is the predominant causative agent in bacterial UTI. That is not true. Remember I said saprophyticus. Once you hear that, this one, in a question, they will tell you in a sexually active female. Young sexually active females are the ones that are most likely going to get a UTI caused by Staphylococcus saprophyticus. But generally, in people who get UTIs, the most common bacteria that you will culture in urine is going to be E. coli. Okay. Then we go to C. Chocolate blood agar is the medium of choice for culture in urine specimen. We don't use chocolate blood agar. Where do we use chocolate blood agar? There's somewhere I mentioned it. Where do we use chocolate blood agar?
Is it for Neisseria species?
Neisseria. Is it mostly used in Neisseria? That was not the answer I was looking for. But Neisseria can be used. The most common one, and you're going to see, is in Haemophilus influenza. Okay? That is where the most common it is used. Okay? So normally in UTIs, as I said, the most common cause of UTI is E. coli. So you're going to use McConkey, and we know that E. coli is lactose fermenter. Okay? You can use CLED as a medium. Choice D, a calibrated wire loop for urinary culture is necessary for the determination of significance of colony counts. So this is the answer. This is true. Okay. Yeah, that's the answer. I don't have any explanations for that. Then the first voided specimen is best for isolation of the causative agent. That's actually not true. What happens is they usually advise that we do a midstream clean catch. Okay, so you void. So because remember, you can have normal flora and we don't want to culture normal flora. So you pee, and then midstream is when you collect the specimen. Okay. Number 6.
Hi, hello.
Yes, yes, yes.
Yeah, on that one, I was just going through the nose, and then somehow the nose which we have, the slice, seem to have something on choice A.
Okay. Yes.
Yeah, choice A, it seems you do get an impression that the answer for that one is A.
What does the note say?
Okay, I'm looking for a special place where this is given,
Because
but I got that.
you see, a UTI, how do people get UTIs? It is not from hematogenous spread. You know what hematogenous spread means? Hematogenous spread means it is spread via the blood. Okay. A UTI is not spread via the blood. So I don't know where you saw it, but maybe you can clip it or take a screenshot, then you can send on the group. We look at it. But as of my knowledge, I've never had a UTI being spread via hematogenous spread.
Okay, I will actually come back to you when I get it.
Okay, no worry. Thank you. Yeah, okay. Number 6. Beta-lactam agents act on the following pathogens except. So the answer here is Mycoplasma pneumoniae. I think I had mentioned this when we did that class. So you have to ask yourself, where do beta-lactam work? Okay. The target for beta-lactam is at the peptidoglycan layer, and that is the cell wall. And you see, Mycoplasma doesn't have a cell wall. You remember we said it lacks a cell wall? Instead, it is replaced by the steroid. Okay. So using beta-lactam agents, these are the penicillins and all those, they will be ineffective when treating Mycoplasma. What is the drug of choice for Mycoplasma? What did we say? Instead of giving the penicillins, And bacteriolactam agents, what drug do we say we use?
Is it isoniazid?
Though not first line. What was the first line?
Doxycycline, maybe?
Yes, so you're thinking of the tetracyclines. Another one? The most common, we use macrolides, okay? Things like erythromycin, azithromycin, clarithromycin, okay? That's usually drug of choice for Mycoplasma pneumoniae. And then yes, you can use tetracycline because all this work by inhibiting the ribosome at different levels, 30S and 50S, okay? We also have, you can also use fluoroquinolones, okay? The rest, they have cell walls, so beta-lactam agents are used. Okay. Number 7. A premature male newborn baby born with low birth weight had a breathing problem and flu-like symptoms. He was immediately admitted to the newborn ICU, NICU. A blood specimen was taken for laboratory examination. Microscopic examination showed gram-negative bacilli. A routine culture was done, and lactose fermenting colonies were seen. Name the possible pathogen. So, do you remember the mnemonic for lactose fermenters? Yes. It was sick. Do you remember sick? Hello?
No.
Okay, you've never heard of that. Okay, let me tell you guys then. It is sick. I think I had mentioned. C is Citrobacter. E, Enterobacter. The other E is E. coli. Then K is Klebsiella. Okay, those are the lactose fermenting bacteria. You will see pink colonies on McConkey. Okay. The rest, these are non-lactose fermenting. So the answer is going to be Klebsiella pneumoniae. Okay. Is that clear? So this is something you have to know, especially during your SAQs or even sport. When you see pink colonies on McConkey agar, those are lactose fermenting bacillus, and you're going to be asked, what are the examples? So the mnemonic is sick. Okay. Let's go to the next one. Number 8. A 45-year-old man comes to the physician due to pain, swelling, and erythema affecting his right leg. He says that he suffered a minor cut to his leg a few days ago while cleaning his garage. Physical examination shows an indurated region surrounding a minor laceration that is draining a purulent exudate. He was diagnosed with cellulitis and started on appropriate treatment. Gram stain of the exudate shows gram-positive cocci in clusters. The organism most likely responsible for the patient's infection synthesizes a protein as part of its peptidoglycan cell wall that does which of the following? So number 1.
Nathan,
Yes.
I think question 8 is different from the question 8 in the paper we did.
It's different.
On my paper, it's repeated. It's a repeated question, and I can't find that question.
Sorry, you see, that's why I never wanted you guys to download as PDF because I was editing. I'm sorry, I wish I had done the final edits. I removed all the repeated questions.
Oh, okay.
Yeah. You see, remember I sent it as a link?
Yeah,
Yeah.
it's okay then.
Is it okay?
Yeah, it's okay. We can continue.
I continue.
Yeah.
Oh, can you see my screen?
Yes, we can.
Sorry, I don't know what happened. Okay, so the organism most likely responsible for the patient's infection synthesizes a protein as part of its peptidoglycan cell wall that does which of the following? So gram-positive cocci, what organism is that? In clusters. Which organism is that?
Staphylococcus.
Staphylococcus, yes. So it can be any of the staphylococcus, but the most likely cause is Staphylococcus aureus. Okay. Now they're asking the organism most likely responsible for the patient's infection synthesizes a protein as part of its peptidoglycan cell wall that does which of the following? So the protein we're talking about here is protein A. Okay. And what does protein A do? So this is a very direct, it's straight from the notes. Protein A binds the FC portion of IgG, okay? And once it does that, it prevents opsonization and phagocytosis, okay? That's just direct from the notes, and you have to know that. The protein A binds the FC portion of IgG, preventing opsonization and phagocytosis. Activates complement. Definitely, a bacteria cannot activate the complement. In fact, it's supposed to avoid the complement system. Okay, causes hemolysis. So Staphylococcus aureus causes hemolysis, but not through protein A. Okay, it produces, remember the toxins produced by Staphylococcus? There's one called hemolysin, which is going to cause the hemolysis. Then cleaves IgA. Cleaving IgA, nope, this is more of Streptococcus pneumoniae, you can remember. Another one was Neisseria. Those are the ones that are cleaving IgA. Streptococcus pneumoniae, Neisseria, and also Haemophilus influenza, but not Staphylococcus aureus. And then interacts with MHC class 2 antigen. What is this? What bacteria does this? Interacts with MHC class 2 antigens. Yes? Which bacteria does that?
Is it tuberculosis?
No. Any other try? So you see, I want you guys to please go back to your notes, because these things are straight from the notes, and this is what they're going to test you. So the bacteria is Staphylococcus aureus, but if you can remember, there were toxins that Staphylococcus was producing. We have the protein A, which is responsible for binding to the FC portion of IgG, causing, preventing opsonization, okay? Then we had, do you remember the toxin, the TSST1, toxic shock like the super antigen? Do you remember it?
Yes.
So that super antigen is
Yes.
the one that is going to bind to MHC class 2 antigens and T cell receptors. Then what is going to happen once it binds? There's going to be cytokine release, and T cell activation. Now that is what is going to lead to symptoms like fever, rash, vomiting, and shock. Okay, the toxic shock syndrome. So that is the pathophysiology of that. And I want you to know that. You see these small, small details, that's what they like asking. The toxic shock syndrome. Then remember the one for enterotoxin, where it causes food poisoning due to the preformed antigens, okay? And then we also have this, the SSS, scalded skin syndrome. So I want you to go look at them. Staphylococcus aureus is very, very high yield, and you have to know that, okay? We clear? Okay, let's continue. The least likely to be a causative agent of bacterial endocarditis. So what's the answer here?
G.
Corynum diphtheria, yes. So there's nowhere where we mentioned corynum diphtheria as a cause of bacterial endocarditis. We have Enterococcus faecalis. So Enterococcus similar to Staphylococcus epidermidis. These ones are nosocomial and they form biofilms on catheters, you know, prosthetic valves and things like that. Then Coxiella species. One thing of Coxiella, remember when we said Coxiella, it is culture, it causes culture negative endocarditis. This is because, remember, Coxiella cannot be cultured the normal way we culture. Okay, so that's why you are going to have endocarditis, but it's going to be culture negative. And Coxiella, this is a zoonotic disease. So if the question is going to come, it's going to be in a scenario of, you know, farmers or people who deal with animals like veterinarian vets. Then Streptococcus pyogenes. Normally, it's not a cause of endocarditis because it causes rheumatic heart disease, but this rheumatic fever can, a sequel of that can be endocarditis. Okay. Then number 10, an outbreak of an infection has been reported in your hospital's ICU. The causative organism is identified as Enterococcus faecium. You are required to present the organism's quantitative antimicrobial susceptibility profile in the next grand round. Which of the following parameters will be of greatest interest to the hospital infection control team? Yes. What's the answer? Yes, you were saying, what is the answer?
I was saying B.
B, yes, good. So how do we answer this question? Number one, you have to know the properties of Enterococcus faecium. So Enterococcus faecium is very notorious for being vancomycin resistant. Okay. So what are these terms that are being used? MIC, MBC, okay? So the increase of the MIC of vancomycin from the previously reported outbreak. So MIC means minimum inhibitory concentration. So when there's an increase in the minimum inhibitory concentration, what does this mean? That there is resistance, and this resistance is worsening. Okay? Are we clear? If there's an increase in MIC, that means that the resistance is worsening. Okay? Now, things like diameter of zone of inhibition of methicillin from the previous 2 years. So if they mention something like Staphylococcus aureus, the answer would be A. Okay? Increase in MIC of amoxicillin, This is amoxicillin is in the group of penicillin, so this is more of things like Staphylococcus aureus. The change in pigment production of the organism on nutrient agar, that is not relevant. That is just a distractor. Increase of MBC of colistin from the previous year. So we don't use colistin. In fact, colistin is more, where did is there anywhere we mentioned colistin as a drug? Mostly in gram-negative bacteria. Okay. Now these terms, MIC and MBC, they're very, very important. Very, very important. And I want you guys to go look at them. So in this case, this question was kind of direct because you already told the diagram, the bacteria, but sometimes, especially in sport, they're going to give you, sorry. Can you see my screen?
Yes.
Yes, as I was saying, the terms MIC, minimum inhibitory concentration, and MBC, minimum bacterial concentration, are very, very important. And I want you to know the definitions and how they're applicable, okay? MIC, minimum inhibitory concentration. From that word, it is the lowest concentration of an antibacterial agent that inhibits visible growth of a microorganism. So the keywords there is lowest concentration. So if you're using 5 grams of amoxicillin and 2 grams of amoxicillin, what is the MIC? So it will be the lower one, okay? The 2 grams one, okay? So it is that minimum concentration to inhibit visible growth, and it has to be visible. Then MBC, which is minimum bacterial concentration, it is the lowest concentration that kills a specific percentage of the organism or the bacteria. And these two are complementary. Okay, so there's a good picture. I'm going to look for it where it has the MIC and MBC because it's very, very common, especially in sport. So take note of that. We continue. The toxin produced by Bacillus anthracis. Select one. Genes for the protein components are found in the chromosome. This is false. So they're not found in the chromosome. They're found in the plasmid. Okay. Lethal factor is a calmodulin-dependent adenylate cyclase. That is wrong. It's not lethal factor. It is the edema factor. You remember? There is the edema factor and the lethal factor. Then C, edema factor binds to the host receptors. That is false. Okay. What binds to the host receptors? The protective antigen. Then D is a combination of lethal factor, edema factor, and protective antigen. That is true. It was made up of these three: the lethal factor, edema factor, and protective antigen. Then is an endotoxin. That is false. This is gram positive, so it is an exotoxin. Okay. You see, if you've not gone through the notes, answering these questions can be quite difficult. So please, 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 please go back to your notes and attempt as many questions as possible because this is direct from the notes. Okay. The Shiga toxin produced by Shigella dysentery is has similar mechanism of action as. What's the answer here?
Is it E?
Veterotoxin produced by enteroinvasive. So look, here is the word enteroinvasive. Is it E? No. Any other answer?
Is it D?
Now you're just guessing now. Now you're guessing.
Sorry. Hello.
Yes, I can hear you.
Yeah, I was looking at it, and I came across this. It said both Shiga and Verotoxin are associated with fetal hemolytic uremic syndrome. So I also thought E could be the answer.
Now, you see, for these toxins, let me send you a summary. There's no like clear summary on these toxins, but let me send you guys. You see, or if it's possible, I can also screen share my screen a little bit. So these are the exotoxins that we're going to be talking about. Now, these exotoxins, they have different mechanism of actions. Now, inhibiting protein synthesis, we have the bacteria relevant here is Corynebacteria, Pseudomonas aeruginosa, Shigella species, and enterohemorrhagic E. coli. But what is the mechanism? For diphtheria toxin, which is produced by Corynebacteria, and Pseudomonas aeruginosa, which is oxytocin A, they both work by inactivating elongation factor EF2, leading to inhibition of protein synthesis. Now for Coryne, it causes pharyngitis with pseudomembranes in throat and severe lymphadenopathy. Then Pseudomonas aeruginosa is going to cause host cell death. Then Shigella, which is the Shiga toxin. You see, for Shigella, it's known as the Shiga toxin, but for EHEC, enterohemorrhagic E. coli, it is the Shiga-like toxin. But they both work by inactivating 60S ribosome by removing adenine from rRNA, leading to damage of GI mucosa, leading to dysentery. Now, dysentery is what we refer to as bloody diarrhea. They also enhance cytokine release, leading to hemolytic uremic syndrome. So I think Steve had mentioned the HUS. Prototypically, EHEC serotype 0157, H7. Then unlike Shigella, EHEC does not invade host cells. Okay, so note, it is EHEC, not EIC. EIEC, that is enteroinvasive E. coli. Then we go to increased fluid secretion. Now, if you are to see here, we have the heat labile toxin of ETEC. This is enterotoxigenic E. coli, and heat stable toxin of ETEC. Heat labile, it overactivates adenyl cyclase through increase in cyclic EMP, leading to chloride secretion in the gut and H2O influx. Heat stable overactivates guanylate cyclase, the cyclic GMP, leading to decreased resorption of sodium chloride and water in the gut. So you see that water is going to remain in the gut, leading to diarrhea. So this is a way of remembering. Bacteria that increase cyclic AMP include cholera, anthrax, pertussis, E. coli. Then Bacillus anthracis, anthrax toxin, specifically the edema factor, increases cyclic AMP as it mimics adenylate cyclase, responsible for the characteristic edematous borders of black Escher in cutaneous anthrax. Then Vibrio cholerae, the cholera toxin, increases cyclic AMP by permanently activating the GS protein. Now you see here, that's why in cholera, they have voluminous rice water diarrhea because it is permanently activating G protein subunit S. Okay. Then we go to inhibiting phagocytic ability. So here it is Bordetella pertussis. What does it do? It activates, the pertussis toxin activates adenylyl cyclase by inactivating GI. GI is the G inhibitory subunit, okay, leading to the whooping cough. Then inhibits release of neurotransmitters. So here are the Clostridium tetanospasmin by Clostridium tetani, Clostridium botulinum. They both cleave the snare protein, a set of proteins required for neurotransmitter release. Okay. Are we clear there?
Yes.
Then you can see my screen. Lysing of cell membranes. This is the Clostridium perfringens. You can see it produces the alpha toxin. Mechanism of action, phospholipase that degrades tissue and cell membranes, leading to myonecrosis, which is the gas gangrene, and hemolysis, double zone of hemolysis on blood agar. Then Streptococcus pyogenes, streptolysin O and S, proteins that degrade cell membrane, lyses RBCs, contributes to better hemolytic host antibodies against toxin ASO used to diagnose rheumatic fever. Do not confuse with immune complexes of post-streptococcal glomerulonephritis. And then super antigens causing shock. There are two, Staphylococcus aureus and Strep pyogenes. For Staphylococcus aureus, it is the toxic shock syndrome toxin, TSST1, and for Streptococcus pyogenes, as I mentioned, erythrogenic exotoxin A, which is responsible for scarlet fever. The mechanism, I think I talked about it. It binds to the MHC class 2 on antigen-presenting cells outside antigen-binding cells, leading to interleukin-1, interleukin-2, interferon, gamma, tumor necrosis factor alpha, which causes shock. And then the TSST1 causes the toxic shock syndrome. Okay, so these are how these exotoxins work, and I want you to remember them. So how they ask in MCQs is they'll ask you, oh, how does this toxin work? Oh, which one works similar to it, or things like that. Okay, so that's a good summary on how it's working. So the Shiga toxin, the answer here should have been enterohemorrhagic E. coli, because they work on the same way due to the Shiga toxin. But now, since we are looking for an answer where it is causing inhibition of protein synthesis, what will the answer be now? From the table, what will the answer be?
B.
B, Corynum diphtheria toxin. Okay. Are we clear?
Yeah, I think, sorry, you can send us that summary afterwards.
I've sent it on the group.
Okay, thank you.
Okay, let's continue. False statement for Clostridium botulinum. Choice A. Exotoxin release interferes with the normal activity of neurons. Is that true? That's true, because we know botulinum blocks acetylcholine, which is going to cause muscle flaccidity, or basically muscle paralysis. Then toxin released in food resists boiling for 1 hour. Is that true? Is that true?
It's not true.
That is not true. Okay. Is capable of colonizing wounds. That is true. Spores in the environment may cause contamination of food. That is true. And then E, exotoxin is the main virulence. That is true. Okay. So the answer is B. Okay, the answer is B. Number 14. So I noticed I repeated number 14. So I put 14A, 14B so as not to confuse you. Then 15, because I didn't want to renumber again. So least likely to be a causative agent of bacterial endocarditis. So it's a repeated question, but different choices. This one was more direct. So what's the answer here? Clostridium tetani. I mean, that is not a cause. Okay. This one causes tetanus. Bacterial pathogen associated with ingestion of preformed toxin in food. What's the answer? What is the answer?
Is it D?
D, Clostridium botulinum. Okay. Same to Staphylococcus aureus. Okay. So this is a very high yield one. Bacteria that cause food poisoning. So write that down. I want you to go check them. Bacteria that cause food poisoning. The preformed toxins are those ones. Staphylococcus aureus and Clostridium botulinum. Food poisoning, are you writing? Food poisoning causes bacteria. I want you guys to know that because it can also come as an essay. They ask you, or the short answered question. False concerning gas gangrene. Choice A, also referred to as Clostridial myonecrosis. That is true. Clostridium perfringens is the commonest cause. That is true. Incubation period ranges between 1 to 7 days. That is true. The presence of aerobic environment is an important step in pathogenesis. So that is false. Why is it false?
Because for Clostridium, we can't be talking about aerobic environment.
It is?
And they are strict anaerobes.
Anaerobes, good. Okay. Strict anaerobes. Then can occur spontaneously. Yes, that is also true. You see, spontaneously means all of a sudden. So especially in people who are immunocompromised, it can occur spontaneously. Okay. Number 16. An epidemiologic study is conducted after a sudden increase in the rate of severe post-operative infections in rural hospital. It was determined that all the patients who developed the infection were operated on with surgical instruments sterilized by a specific autoclave. The machine was found to be inadequately sterilizing the equipment due to malfunctioning, limiting the temperature to 100 degrees during the 15-minute cycle. These patients most likely developed infection from which of the following bacteria? So, I mean, it's a very long question, but the question is asking, which bacteria is resistant to temperatures up to 100 degrees? Because they're saying the temperature of 100 degrees. So from the question, that is the question they're asking. Which bacteria do you think from these ones mentioned are resistant to temperatures up to 100 degrees?
D.
D, good. Clostridium perfringens. Why?
Because that one contains spores, and one of the functions of spores is they are resistant to heat or adverse conditions.
Very good.
Yeah, I was saying that
Yes.
those strict anaerobes, Clostridium, they have spores, and with the presence of spores, they would resist adverse conditions, which would include heating.
Yes. So heating, things like chemicals, temperatures, high temperatures, heating, and things like that. Okay, due to the spores. So the spores are their survival mechanism, and they are going to germinate and cause infection. Okay, so that's very good. Then we go to number 17. It is asking Listeria monocytogenes. Okay, hope you remember that bacteria. Is it a strict aerobe, facultative? No, you see, sorry, this facultative should not be there. The question should read strict aerobe. So it is not a strict aerobe. It is a facultative anaerobe. Okay. Causes granulomatosis in Infanseptica. That is true. That is the answer. Strict man pathogen. Basically what that means is it zoonotic? So Listeria is zoonotic. It can affect animals. Okay. Gram-negative Coccobacilli, no. Listeria is gram-positive. Form spores in nutritionally deficient media, nope. Only spore-forming. What are the only two spore-forming bacteria that we said? And we will be seeing that.
Clostridium and bacilli.
And bacilli, very good. Actinomycetes israeli and Streptomyces somanilensis possesses the following characteristics in common. Normal flora in mucosal surfaces. Actinomyces, yes, it's a normal flora, but is Streptomyces normal flora? That's not true. Strict anaerobe. Actinomyces, anaerobe. Streptomyces, aerobe. Strict aerobe, so Streptomyces is an aerobe, this is an anaerobe. Acid-fast filamentous bacilli, that is true. Okay, filamentous gram-positive. No, sorry, this is false. It's not acid-fast. Acid-fast is Nocardia. Filamentous gram-positive microorganism, that is the one that is true. Acid-fast is Nocardia. Which other bacteria is acid-fast? The common one?
TB.
TB, yes. Okay. So they like asking such questions. Maybe one or two, you won't see a lot of questions, but they'll ask actinomyces, streptomyces, and nocardia. This is because they're the filamentous or branching filaments. They cause branching filaments. What I want you to remember is actinomyces is an anaerobic. It does not require oxygen. Streptomyces, nocardia require oxygen. And then this one is not acid fast. Nocardia is acid fast. Okay, just that. They won't ask a lot.
I have a question.
Yes.
You said actinomyces are anaerobic. Are they strict anaerobes or facultative?
Actinomyces are strict anaerobes.
Okay, thank you.
Okay. A 6-year-old boy is admitted to the hospital because of 8-hour history of fever, headache, and nausea. Administration of intravenous fluids and empiric antibiotics started pending culture results. A lumbar puncture was done. Gram-negative bacteria was seen. During the next 24 hours, he develops bleeding from his gums and all IV puncture sites. The most likely cause of the patient's new symptoms is which of the following infection-related complications? So before you answer this question, you have to ask yourself, what bacteria is this? What bacteria is this?
Neisseria meningitis.
Neisseria meningitis. Very good. Why is it that? Because these are symptoms of meningitis till here. And then gram-negative bacterial seen. So see, the most common cause of meningitis in children is strep pneumoniae and then Neisseria meningitidis. Now, what complication is this? Considering you're bleeding and yes, there is bleeding and meningitis. What complication is that?
It's, number A, which is adrenal hemorrhage.
It has a name.
So,
You see, it is somewhere there in the notes. I don't know if you can remember it. It is known as Waterhouse, Friderichsen syndrome. So basically what is happening is the Waterhouse Friderichsen syndrome is when you have adrenal hemorrhage, okay, leading to symptoms of bleeding. So you see the bleeding gums and IV puncture sites, plus a history of meningitis, okay? There is adrenal insufficiency. You know the adrenal gland, the ones that sit above the kidneys, okay? So once you see something like that, this is Waterhouse-Friderichsen syndrome, Waterhouse-Friderichsen syndrome, okay? Who got this question?
I got it after I was through looking at the nose.
Yeah, it's not an easy question. Like it's something you have to know, you know, like if you read, if you have surface knowledge, you won't get this question correct. You will just guess. I know most of you maybe took E, toxic toxic epidermal necrolysis, 10. What is the most common cause of this? Choice E. Skin infections. What are the bacteria that cause skin infections?
Staphylococcus aureus.
Staphylococcus aureus. Here, another one. It's a another one. Streptococcus pyogenes. Okay, those are the two most common causes of skin manifestations. So we saw a question on cellulitis that the answer was Staphylococcus aureus. Okay, those are the two most common causes of skin infections. Okay. Number 20. Which of the following acid-fast rod bacilli can take up to 10 years for its growth in host cells and causes skin infections? Yeah, so this is very direct. If you see acid-fast rod bacilli, that is Mycobacterium. So which one? So what's the answer here? What's the answer here? Answer?
Is it E?
Yes, leprosy. You see Mycobacterium leprosy. For those who have read the Bible, do you remember a disease called leprosy? These days it's uncommon. I've never even seen it. But we learn about it because it is in the family of Mycobacterium. So if you remember, if you've read the Bible, Jesus heals the 10 lepers. So these people had leprosy caused by Mycobacterium leprae. So for you to remember this, you can remember that. And leprae, it causes the skin infections. Okay. In pharmacology, you'll be told there are two types. We have the lepromatous and tuberculoid, where the different drugs are used, dapsone, rifampicin, and things like that. Okay. But yeah, if you see skin infection, acid fast, Mycobacterium leprae. So false about Neisseria meningitidis. Choice A, ferments glucose and maltose. Is that true? Is choice A
Yes.
true? That is true. Remember, Neisseria meningitidis, glucose and maltose. Neisseria gonorrhoeae, glucose alone. Ion sequestration mechanisms are an important virulence factor. Yeah. That is true. Okay. You see, Neisseria meningitidis, it requires iron. Why? Because it has ion binding proteins. So ion sequestration mechanism is very important in its virulence factor. Then shares 80 to 90% genomic homology with Neisseria gonorrhea. That is true. Serogroups are classified according to polysaccharide capsular antigen. That is also true. Oxidase negative, that is false. They're usually oxidase positive. Okay. Selective medium for Neisseria gonorrhea. This is direct. Diamartin. Okay. I hope everyone got this correct. Where do we use Skiros? Skiros medium, where do we use it? Have you heard of Skiros? Hello?
I saw it somewhere, but I can't remember.
It is found in Campylobacter. Thiosulfate, citrate, bile salts, sucrose. TCBS, this is very common. You should be knowing this. It appears yellow. TCBS, thiosulfate, citrate, bile salts. Vibrio cholera, okay? Lowenstein Jensen.
Is it Bodotera?
LJ. LJ, mostly TB.
Which one is that for Bodotera toxin?
Bordetella. Remind me in full. Abu, say it in full. Bordetella pertussis.
That which causes whooping cough.
Oh, yeah. Sorry. You know, I usually confuse Bordetella and Coraini. Bordetella is the whooping cough one. Bordetella.
Sorry?
Let me check. That one I've forgotten. Bordetella. But I know for LJ, Lowenstein Jensen is for TB. Bordetella, I'm seeing here, they're saying charcoal. I'm not seeing it. Unless you guys will see it, then you tell us. The black one, Regan Low. It's called Regan Low, the black one. Okay. Regan Low agar. Okay. Number 23. Human infection of which of the following organism occurs through inhalation of bird droppings? So I remember I said this when I taught it recently. This one, Sitaki. Okay. Oh, what has happened? Chlamydophilia. Sorry. What is happening? Chlamydophilia Sitaki. You remember parrot dropping? I don't know what I've done. Chlamydophilia Sitaki. Okay. Yeah, this is the direct one. A heat labile toxin secreted by E. coli stimulates. So if you go back to that table, check the answer. What does it stimulate?
C.
CMP. Then heat stable, cyclic GMP. Okay. Which of the following gram-negative bacilli are not motile? Yeah, this is also direct. Shigella is not motile. The rest are motile. Not useful in the lab diagnosis of Rickettsia species. Who got this question? This is also, it's not an easy one. Who got this? Number 26. There's tissue culture. Vancomycin containing blood agar, GEMSA stain. So it's not GEMSA, latex agglutination, OX19 strains of Proteus vulgaris. So the principle of answering this question is you have to remember Rickettsia is an intracellular organism. Okay? And it is obligate intracellular, meaning it lives inside the cell. So the answer is this blood agar. It cannot grow outside the cell, so you cannot use a blood agar. Okay. Then general characteristics of Yersinia pestis and include all the following except so bipolar staining, that is true. That is a must. A little bit, let me go get that. So we were talking about Yersinia pestis. So the answer here is gram-negative Coccobacilli, that is true. Capsulated, that is true. Bipolar staining, that is true. Facultative intracellular, that is true, because they survive in macrophages. So here the answer is strict aerobic. They are anaerobic. Okay, they are facultative anaerobes. Bacterial pathogen associated ingestion of preformed toxin in food. So we have done this. It is Clostridium botulinum. This is a repeated question. The following are important of Vibrio cholerae except. So number one, highly motile. I mean, that is very true. Remember, they have that tail or the flagella. Okay, and it's a single polar flagella. Non-invasive pathogen, that is true. Remember, we said it causes the rice water diarrhea via the exotoxin. And how does the exotoxin work? It increases cyclic AMP by activating the G sub S subunit, permanently activating it, causing rice water diarrhea, and it is very voluminous. Produces cytochrome oxidase enzyme. That is true. Basically, what this means is it's oxidase positive. Occupies habitats of high acidity. That is false. You see, for Vibrio cholerae, it does not do well in high acid areas. That's why in the stomach, the acid in the stomach is going to destroy the Vibrio cholerae. That's why it requires very many species for it to be infectious. Okay? So Vibrio cholerae does well in alkaline situations. So for people who are taking PPIs are proton pump inhibitors. Proton pump inhibitors reduce the acid level in our body. They are at an increased susceptibility of getting Vibrio cholerae. Gram-negative curved rods, yes, so we usually call them comma-shaped. Okay. Then finally, gastrointestinal tract pathogen that causes infection by adherence to intestinal mucosa. Adherence means to stick. Okay. It is EPEC, enteropathogenic E. coli. Okay. So the rest, Salmonella, that one invades the mucosa, especially the M cells in Peyer's patches. You've done Peyer's patches in physiology. M cells of Peyer's patches. Then Campylobacter jejuni, that one causes invasion. Bacillus cereus, the preformed toxins, you remember? The two types, emetic and diarrheal type. And then Vibrio cholerae. This one does not cause adherence. It is toxin mediated. Okay. So thank you so much. Any questions? We'll continue with the other section on Tuesday. Unless you have any questions, I think we can finish there.